Good afternoon, everyone. I hope you guys are having a good day. So today we're going to go ahead and review chapter 3. Okay, so in chapter 3, we're going to be looking at protein structure and function. Okay, so the very first thing here that I have on the screen is the different functional groups that we talked about in chapter 2, right? So the very end of chapter 2 had a functional group. Remember, a functional group is like a trend. It's going to be a chemical formula that is going to have very specific characteristics, right? So for example, here on the screen, I have an amino acid, the amino functional group. I'm going to go ahead and skip through some of these slides as we'll talk about them in lecture. But today I'm going to give you guys a brief overview of chapter 3. So why are proteins important to talk about? Okay, so proteins are very important because they each have a very specialized function, but proteins in general are going to be diverse in function. Okay, so almost all of the functions in your body are going to be using proteins to go ahead and complete them. And today we're going to be talking about amino acids, at least for the first half of the chapter, and that is because amino acids are the building blocks of protein. Okay, so when we get into the structure of proteins, we need to look at the structure of amino acids. Okay, so there's about 20 amino acids that we use to make proteins. Okay, and amino acids are going to be composed of a central carbon atom that is bonded to a hydrogen atom. Okay, so like if you look down here at the bottom, you can see that we have the structure of an amino acid here, right? So we're going to have that initial carbon that's always bonded with this hydrogen atom here, okay? Then you can see that we're going to have different functional groups, right? So here on the left, we have the amino group, right? So this is one functional group. And then here on the right, we have the carboxyl group, which is the second functional group, okay? Please note that at the bottom we have what's referred to as an R group, okay? An R group is going to be like a spot saver for other kinds of side chains, okay? So this is going to be like the area where those 20 amino acids that we just talked about are going to be different, okay? So all 20 are going to have the amino group, we're going to have this centralized carbon, and we're all going to have the carboxyl group, okay? They're all going to have a side chain, this R group here. The difference is that every single R group will look different. Okay, so skipping through, right, we have some examples here of additional structure, right? Remember that I said we have that central carbon, and this is going to be resulting, these structures here, the amino group and the carboxyl group, are going to be helping with amino acids to stay in solution and to make amino acids more reactive. Okay, so these are having partial charges. Remember that we are having a polar covalent bond here. That's why you have these partial charges, and these partial charges are going to go ahead and cause these different characteristics of this amino acid. Okay. So like I mentioned, right, every single 20 of these amino acids will have a different unique R group, okay? This is also referred to as a side chain, okay? And this is going to be attached to that central carbon, okay? Side chains can be grouped into 3 types, okay? We can either have charge groups, which are going to include our acidic or basic side chains. We can have uncharged polar, and we can also have non-polar side chains as well. Okay, so here we have the different amino acids all kind of characterized by the different types of side chains, right? So here first we have the the electrically charged side chains, right? So we have 2 examples of acidic, 3 examples of basic. We have the polar side chains that are going to have these here in the pink. And then we have, most of them are going to be under the non-polar side chains, which will be here in the green. Okay, I'm going to kind of skip through here as this is just a general overview of the whole chapter. We'll go in this much more in depth when we go through in class. Okay. Okay, so why exactly are proteins important, right? So I just mentioned earlier that proteins are going to have very different specialized functions, right? And proteins are referred to as a macromolecule, okay? Macromolecules, macro meaning a little bit larger, molecules, right? We have several atoms bonded together. So these are going to be large molecules that are made up of smaller subunits. Okay, this subunit is referred to as a monomer. Okay, so like one part. Remember I gave you guys when we first initially talked about monomers, I gave you the example of like a Lego piece, right? This is going to be that small little square that would build up to become the actual item that you're building, like the dinosaur or so on. Okay, so here we have the monomer for a protein. This is going to be amino acids. Okay, so the monomer is going to be amino acids for proteins. And all of these monomers are going to be linked together, and they're going to be, this term is referred to as polymerized, okay, and they're going to form a polymer, so it's going to have many parts. So in this case, right, amino acids is the monomer, proteins is the polymer. Okay, so we have the process of how we're going to have monomers be polymerized, right? And this is through condensation, or what's referred to as a dehydration reaction, okay? So dehydration, right, when you're dehydrated, that means that you're going to need water, and so that means that there was a loss of water, right? So the exact same thing here is happening. Condensation reactions are resulting in loss of water molecules, okay? So we're going to have these amino acids, and this is where this we're going to see that we're going to have one polymer here and the second polymer over here, and you can see that these two here components will separate and become water. So at the end, you're going to have a structure similar to this with the monomer now added to the polypeptide chain, the polymer here, and now we have the release of water. Okay, hydrolysis is going to be the reverse reaction. So this is going to be when we break up a polymer into monomers, right? So here again, you can see that we have the exact same polymer here. This is the monomer that we had just added. If you separate that bond, you'll see that we have a water being separated and then added to each ends of that polypeptide chain. Polymerization always requires energy, okay? So monomers would not self-assemble into a polymer, right? So this is basically saying we can't just spontaneously make proteins, right? It's going to be a process where energy is required, okay? Amino acids will polymerize when we have bonds that form between the carboxyl group and one of the amino groups. Okay, that's how we're going to have the two monomers kind of like form together a bond. So you can see here we have a carboxyl group of one amino acid, right? And then we have the amino group of another different amino acid. And here's where you can see clearly where you would have that water formed, right? So you have this water and this oxygen here. And then again, at the end, you're going to form a peptide bond where you have the loss of water. Okay, here's the water. Okay, so when we're talking about peptides, they're going to be forming, peptide bonds are going to be forming this backbone, okay? And we have two distinguishing characteristics, okay? So R group orientation is going to be how the side chain extends out and can interact with each other with water, okay? So basically, how is this going to be able to interact with each other? How is it going to be able to form? And then second, we have flexibility, so single bonds on either side of the peptide bond can rotate, okay? So meaning that we're going to have the ability to kind of flip around to change the orientation of that R group. Okay, so again, I'm just briefly going through all of the topics for today, but you'll see here that we have different types of peptide bonds. So polypeptides or polymers that have a chain fewer than 50 is referred to as an oligopeptide. Okay, so this means few. Okay, so this means that it's going to be a chain of 50 or less. Okay, a chain that is more than 50 is going to be a polypeptide, so many peptides. Okay, but these are both describing proteins. Okay, so proteins are the complete functional form of the molecule, but these oligopeptides and polypeptides are essentially proteins. So one of the Okay, so there's different structures for proteins, and this is very important for their functions. Okay, so the very first type of structure is primary structure. Okay, so protein primary structure is going to be a unique sequence of amino acids. Okay, so this is like the amino acid sequence that we're talking about. Okay, that's the primary structure. The number of primary structures can be basically limitless, and that's why, like I was mentioning, right, proteins are going to have such diverse functions, and they're going to be able to perform a lot of different functions in your body because of the diversity in the primary structures. Okay, so 20 types of amino acids are available, and proteins can range from either 2 amino acids all the way to more than 10,000. Okay, so that's why, for example, we can have extremely diverse functions in proteins. The primary structure is the fundamental, is like component for all of the other structures. So this is basically going to tell you what exactly or which type of protein we're creating here. Okay, so we also have additional structures like secondary, tertiary, and quaternary. Okay, these are going to be additional, more complex structures. Okay, so primary structure is linear, and it's going to be the amino acid sequence. Okay, the secondary structure is going to be formed between hydrogen bonds. So you can see that now we have that primary structure being bonded within itself. Okay, so here we're going to have the carbonyl group and one of the amino acids bonding. So like amino acids are going to be bonding with another amino acid through hydrogen bonds. Okay, remember that hydrogen bonds are the weakest type of bond, and they're always happening between an oxygen and a hydrogen, and it has to do with those partial charges, right? So like the electronegativity, it has to do with the attraction of those opposites. Okay, so we have different types of secondary structure. We have the alpha helixes, or we have the beta pleated sheets. Okay, the alpha helixes are going to be these kind of twisted ones that kind of look like DNA, right? And then the beta pleated sheets are the ones that are kind of like sheet-like, where they're not like twisted, they're kind of just bonded to each other, right? So this is what the structures would look like in terms of the secondary structures with the bonds, the hydrogen bonds, but they're often depicted in this kind of view. Okay, so this is what an alpha helix would look like in diagrams, and this is what beta sheets look like. Okay, so when you see these two structures, know that this is what they're representing. Okay, so again, this would be the primary structure here, right? And then because we have hydrogen bonding, it forms into the secondary structure. Okay, next we have tertiary structure. Okay, and tertiary structure gets a little bit more complex, right? So we have 5 important types of R group interactions. Remember, the R group is going to be that side chain, and hydrogen bonds are going to form between those polar sides to those opposite pole partial charges. Okay, so think of it as like now we have those beta sheets, those alpha helixes, but we're going to have the additional interactions between the partial charges, right? So we have hydrophobic interactions. Remember, hydrophobic means that we have water hating, right? So this is going to be forcing water forces hydrophobic side chains together. Okay, so this is kind of like what the protein would look like here, but because it's hydrophobic, what's going to happen is that it's kind of going to be bonded within itself, kind of collapsing. Okay, next we have tertiary structure. Okay, well, I guess we're still talking about tertiary structure, but here we're looking at a different types of bonds. So this is a disulfide bond. Okay, and this is going to be forming bridges. So it's 2 sulfur bonds that are 2 sulfur atoms bonding together. Okay. We have ionic bonding that's going to be forming as well. So remember, that's complete transfer of electrons. Okay, so again, right, we have different examples here. So we have beta sheets and alpha helixes all throughout this one protein, right? Those are the secondary structures. And we're going to have tertiary structures kind of combined. So you can see that they become more complex, and it has to be dealing with bigger proteins, right? So the interaction is going to be shown there. Okay, And then lastly, we have quaternary structures, which are going to be 2 or more distinct polypeptide subunits that are going to be bonding together. So essentially, we're going to have these polypeptide subunits, so different sections that are coming together to become one structure, right? So we have one alpha here, and then we have the beta 1 and 2, okay? So here's a good example, like with the array diagram, what all these different types of bonding structure do, right? So protein structure, we have the primary, right? Remember, that's like the amino acid sequence. Secondary is either going to be an alpha helix or a beta sheet. Tertiary is going to be that bonding of the interactions, right? Those are like becoming, those hydrogen bonds are kind of collapsing within it, right? Where we have those partial charges attracting. And then here we have several of them coming together, right? So we have, this is like one protein. Now we have the blue one, the light blue, and the darker green. They all come together for the quaternary structure. Okay, so once we talk about structure, we're going to be talking about a concept referred to as folding. Okay, folding is essentially those structures adequately being formed. Okay, so that means the secondary, the tertiary, the quaternary structures all being formed properly. Okay, and this is extremely important because these are going to be allowing for proper function. Okay, so protein folding is often spontaneous. So as the protein is being made, it's going to be properly folded. Okay, but we have a process referred to as denatured, and this is essentially unfolding, and it's going to be unable to function normally. Okay, so here we have an example of a protein, right? You can see we have a secondary and tertiary structures here, and then we have denaturing happening where now we kind of lost the secondary and tertiary structures. Okay, so protein shape is flexible, so each protein has a characteristic that's going to be folded. Okay, but the folding is going to have a very specific function. So many proteins have disordered shape when they are inactive, but when we have active proteins, it needs to be in its proper folding to have an active conformation, right? So it's going to allow it to perform its essential function. Okay, so we have what's referred to as either misfolding or like that denaturing. Okay, this is going to be when those proteins do not properly function. Okay, this can become an issue specifically because misfolding can become infectious. Okay, so we all know viruses are not considered living, they're very scary, right? Well, there's one thing that's scarier than viruses because it's still not living, and those are prions. Okay, so these are going to be improperly folded proteins, and what happens is they can cause disease. Okay, so this is essentially, right, what the normal protein is supposed to look like, but here now you can see that it's still the same structure. We have the same amino acids sequence, right? We still have the same primary structure, but what happened is that now we have, instead of having alpha helixes here, we now have beta sheets, and because of the structure, this pyron becomes infectious. Okay, so essentially think of it as these are going to be big issues, primarily having to do with the brain. Okay, so like I don't know if you guys have heard of mad cow disease, but mad cow disease is caused by pyron. Okay, essentially what happens is that you're going to have normal brain tissue, right? So this is a diagram of our brain. You can see that we have all these different proteins and everything is working properly, right? Here in the blue, these are going to be a pyron, so it's an abnormal shape. So you can see that this is different shape than these purple ones. And what happens is that we're going to have them kind of only bonding with each other. So you can see that they're bonding here. And what's going to happen is that we're going to have a pyron chain. And so these large structures then prohibit proper brain function. Okay, so like I mentioned, right, this is going to include mad cow disease. Okay, so a pyron is going to be an infectious protein, and this is going to allow for misfunction of the brain. Okay, so it's like essentially kind of forming like clots, but it's not involving blood. So it's these big protein structures that are going to prevent the brain from properly functioning. Okay, another additional pyron disease is referred to as the chronic wasting disease. Okay, scabies is also one of these. Okay, So these, although proteins have a lot of necessary functions in our body, they can also cause disease. Okay. So now that we talked about what happens if they don't perform their functions normally, let's talk about some of their actual functions. Okay, so this is something that even the pre-AP class is learning because this is super important, but proteins have critical tasks throughout the body, and especially in our cells. Okay, the first and most common example of protein function is referred to as the catalyst. Okay, these are referred to as enzymes as well, but this is going to be the process of speeding up chemical reactions. Okay, so this is when we're going to see the chemical process kind of go upward, right? And we're going to be able to produce more of chemical reactions than we normally do. Okay. Next is something that's very common and talked about currently, right? And that is because proteins are antibodies, okay? And antibodies attack pathogens within your body, right? So that's the function of a vaccine, right? A vaccine would introduce the infection prior to getting sick, so like you don't have to get sick, and it will teach and show your body how to create antibodies without actually being infected, right? So right now it's a very common topic because of the pandemic, but antibodies are going to help attack pathogens. Okay, so I like to think of them as our natural medicine against infection. Okay, next we're going to have very specialized proteins that are going to help move cells or molecules within the cell. Okay, so this has to do with movement. We also have proteins important for signaling, so it's going to be signaling between cells. We have proteins that are going to be important for structure of cells as well as our body structures. Okay, and then transport, so we're going to have molecules that are going to be able to enter and exit the cell solely due to proteins. Okay, so here's another complex example of a polypeptide chain, right, or a protein. Okay, so we're going to talk a little bit, I'm going to emphasize a little bit about enzymes because enzymes are really important when we talk about proteins. Okay, so like I mentioned, right, enzymes are going to be a type of protein and they're going to be a catalysis, meaning that they're going to speed up chemical reactions. Okay, so enzyme is a protein that functions as a catalyst. Okay, so we're going to have an enzyme and a substrate. Okay, substrates are going to be reactions. So we're going to have a enzyme catalyzed reaction occur, and the substrate is going to have something changed from it, right? So it can either be broken up or it can be put together, okay? The location of an enzyme where substrates bind and react is referred to as the active site, okay? In class, when we go over this, we'll go ahead and spend some more time, but this is very important. It's going to allow for less energy at the beginning of a chemical reaction, and that is exactly how we're going to speed up the reactions. Okay, so this is the